Namaskar दोस्तों, welcome to channel Toxicology on board. Today in this video we'll be discussing another DABT topic, that is fumonisin, which is a type of mycotoxins. So let's directly dive into the question. The question is taken from ABT, that is American Board of Toxicology recertification question paper, year 2000. Part A. It consists of three parts, 50 marks question. These are old type of questions. Nowadays this type of pattern is not there, but yet from these questions we'll try to learn the concepts. Okay, so let's directly see the question. Fumanescins are secondary metabolites of Fusarium moniliforme, Fusarium proliferatum, and other Fusaria that commonly contaminate corn. So we need to identify this fungal species. Okay, so we have three different strains, that is moniliforme, we have proliferatum, and we have other Fusaria species which are going to contaminate the corn. So we need to identify these mycotoxins are reported to be associated with. So we need to identify the characteristic feature of this type of fuminotoxins. Okay, so we have option A, that is equine leukoencephalomalacia. Option 2, we have porcine edema, that is pig edema. First one is related to horse, second one is related to pig, third one is related to rat, that is rodent hepatotoxicity, rat liver toxicity. Fourth one is human, that is esophageal cancer. Okay, so right option will be in the green text, that is option E. All the four options. It is going to affect horse, it is going to affect pig, rat as well as human. Okay, so one by one, each and every option will try to read briefly, so that we'll be able to have some awareness regarding these particular options. So we have option number one, that is equine leukoencephalomalacia. As the name indicates, equine is something related to horse. Horse tail, that genus will be there, that is called equine. And encephalomalacia, something related to head. So we'll see. Equine leukoencephalomalacia, that is ELEM. Another name for this particular state is this condition is moldy corn poisoning. It is fatal. Acute neurological disease. It is going to affect your CNS, that is brain, in the case of horse, and caused by how whenever this horse is there, no, when it is eating this contaminated corn, the corn which is contaminated with particularly this Fusarium moniliforme, in corn, which produces fuminotoxin. So on this corn which is contaminated with this fungal toxin, mycotoxin, Fusarium species, particularly moniliforme. So whenever this horse is eating this contaminated corn, this particular equine leukoencephalomalacia will be there. So what are the basic symptoms means? Um, you will see depression, head pressing. This horse, they will try to press their head towards the wall or towards the pole, circling, blindness, rapid death within after consuming within 48 to 72 hours, like within 2 to 3 days that horse will die. Okay, so as such there is no specific treatment, but prevention is the best thing. Avoid contaminated consuming this contaminated corn food. Try to avoid giving this type of contaminated corn to the horse. Okay, let's see what is second option, that is porcine edema. Coming to porcine is something related to pig. You have to remember in that way, and edema is related to inflammation, swelling. Okay, so porcine pulmonary edema means something related to lung. So whenever these pigs are there, barn in the area where the pigs are there, cattle is there. Whenever they inhale the dust from this particular contaminated corn, which is contaminated with fungal toxin, then those particles will enter into the lung and it will cause pulmonary edema. So we'll read about what is this PPE, that is porcine pulmonary edema. It is a fatal acute swine disease related to pig, okay, caused by ingestion of fumonisin mycotoxin produced by this particular strain, that is Fusarium verticillioides. Nowadays it is formerly it is no, it was called as moniliforme. In equine encephalomalacia also same strain we have seen in contaminated corn. This whatever this porcine pulmonary edema is there, no, it will cause us severe pulmonary edema, lung, okay, heart failure, death within 3 to 6 days. Earlier equine encephalomalacia 2 to 3 days. Here, within 3 to 6 days of consuming this contaminated corn, more than 100 ppm fumarocin, whenever this particular contaminated corn contains, it will lead to death within 3 to 6 days. Then let's see what are the symptoms are there. Severe dyspnea, since it is related to respiratory, difficulty in breathing will be there. Cyanosis. So whenever you will come across this word cyanosis, you have to correlate and read about cyanide poisoning. You have to read about carbon monoxide poisoning. So always you have to correlate whenever the cyanosis word is coming. No, read about that one. Coming back to porcine edema, other symptoms are weakness, up to 100% mortality in the affected pigs. Okay, so let's see what is option C. So till now we read about horse, then we are reading about pig. Now third one we are discussing about the third option, that is rodent hepatotoxicity. Rat liver toxicity. How the rat, whenever they're consuming or they're affected with this fungal toxin, how it is affecting the liver toxicity. So what is the pathway which is going to interrupt, that we will see. Fumonisin FB1. See, in the fumonisin also various classes will be there. Particularly FB1 is the one which is having this severe adverse effects. So we have, it is a potent mycotoxin that induces liver toxicity, hepatotoxicity in the case of rodents. Rodents means mice and rat, particularly mice, through inhibiting ceramide synthase. So you will come across this one in the biochemistry. Lipid metabolism will be there, fat that synthesis will be there. No? In that you will come across this particular one thing, ceramide synthase. So this is going to inhibit, this particular toxin is going to inhibit this fatty fat metabolism. Okay, ceramide synthase, whatever the in biochemistry you'll come across, no, glucogenesis, glycogenolysis, all those type of, in that lipid synthesis cycle will be there. In that particularly step, ceramide synthesis it is going to affect, leading to sphingolipid metabolism disruption. As I told you, know, various steps will be there in the lipid synthesis. Ceramide is also you'll come across sphingolipid also. Oxidative stress, thereby it will lead to oxidative stress. Then it will cause apoptosis. Two types of death will be there: necrosis cell death, apoptosis cell death. So it is causing apoptosis cell death, and finally it leads to liver fiber and liver fibrosis. Okay, so whenever it comes to liver fibrosis also, whenever you are reading about liver toxicity, you have to see how certain agent is or chemical is affecting acutely on the liver, then chronic, how it is going to fibrosis and completely liver failure, end stage liver failure, or ending up with liver cancer. So that stages you have to remember. Okay, so coming to FB1, fumonisin B1, no, it acts as a liver carcinogen. So now you understood why I talked when hope you understood why when I mentioned about the various stages. Eventually, after fibrosis, it will lead to cancer. So this FB1 is also causing liver cancer. Okay, and high doses are causing hepatocellular hypertrophy, that is increase in the cell size of this liver cells. Okay, increased serum ALT and AST levels. So these are the liver biomarkers. Whenever you have a liver failure, you are using these two biomarkers, ALT as well as AST. Okay, so there will be increase in this serum ALT and AST levels. So you can pause the video and you can see this particular pathway, how it is affecting ceramide synthesis and sphingolipid metabolism. So till now we have discussed about horse, then pig, then rat. Now we are discussing about human. How it is going? These fumanosins are going to cause esophageal cancer in the case of humans. So fumonisin B1. Still we are continuing discussing fumonisin B1. Why? Because it is a potent mycotoxin. It is a toxic byproduct of fusarium fungi found in the maize or corn. These terms you can use. Classified as group 2B carcinogen. This is very important. So here you can pause here and try to remember, or earlier if you have read IARC, that is International Agency for Research on Cancer. IARC is the body from the US only. So they take certain class of chemicals where you find that they are having potential for carcinogenicity or they are causing cancer. They will classify that cancer, that particular chemical into 5 types will be there. So this particular fumonisin will fall under group 2B type of carcinogen. So it is possibly carcinogenic to the humans and is strongly linked to human esophageal cancer cells. So this is a particular part in the case of digestive tract where it is causing esophageal cancer, right? Then we'll see what is happening upon chronic ingestion of this contaminated corn in the case of human. Chronic ingestion of this FB1 contaminated corn induces cell proliferation. So first step in the cancer is cell proliferation, rapid multiplication of the cells. Then migration of this cancerous cells to other regions in the body. Okay, and DNA damage in the esophageal cells. So there will be irreversible type of damage, and not only to that particular generation of that cells, but to the subsequent generations, right? So that is DNA damage in the esophageal cells, leading to tumorigenesis, means um, leading to more number of cancerous cells through pathways like they're going to inhibit HDAC inhibitor, phosphoinositol 3 and AKT pathway. These are the different pathways will be there. Okay, so these pathways are disrupted and it will lead to cancer. So this is the mechanism. So being a toxicologist, you need to read about mechanism of toxicity also. Certain times they will ask, not too much I have seen in the ABT, DABT question paper, but as a toxicology, you should know the mechanism. Okay, then we will discuss something. So till now we have discussed about four options. Now we will discuss something related to fumonisin only in the case of Clayson textbook. As I told, two textbooks. One is Clayson, big, the larger volume, that is 1500 plus pages textbook you can read, or I am sticking to smaller version, that is around 600 to 700 pages textbook will be there. So under chapter 27, food toxicology, this will come under. It is this fumonosins or mycotoxins, plant toxins are a part of DABT syllabus only. You can go back and check the brochure. Okay. So on page number 1349, sorry, this is taken from the bigger textbook only, that is Clayson 9th edition, page number 1349, chapter number 27, food toxicology. So we'll very quickly go through what are the important things that we can learn and which can be useful for our exam. So fumaroles are produced by a number of fusarium species. So what are those species? We have Fusarium verticillioides, that is formerly it is known as Fusarium moniliforme, Fusarium proliferatum, and Fusarium nygamai. So these are the three things. In that also frequently what we are coming across is moniliforme. Okay. Major species of economic importance are Fusarium verticillioides, or formerly it was moniliforme, because it grows on corn in both vegetative and reproductive tissues. So this is very important. Okay, this Fusarium verticilloides is present in virtually all corn samples. Okay, and it is you can see in different regions in Africa, America, so many geographically this disease has been seen, including in India also. We'll discuss one case study. Most strains do not produce a toxin. So the presence of fungus by default, it doesn't mean that it is having fumonisin. Okay, most abundantly, it is present in the one is fumonisin B1, which we have previously seen. All the options also we have come across fumonisin B1 only. Fumonisin affect animals in different ways by interfering with sphingolipid metabolism. As I told you, they are going to affect the fatty metabolism or lipid metabolism. Okay. Then these fumanosins, they are going to cause leukoencephalomalacia, that is hole in the head syndrome. That is in the case of horse, whenever the horse who is taking this particular contaminated corn with fusarium species, they are going to get this hole in the head syndrome. Here you can see the brain. Okay, you can see the hole here in equines as well as in the rabbit. In our option only equine was given. Okay, pulmonary edema and hydrothorax in the case of swine, that is in the case of pig. Second option we had liver, that is hepatotoxic and hepatocarcinogenic effects. So liver toxicity, cancer, that also we have seen fourth and third and fourth option. Apoptosis in the case of liver and the rats. The mechanism, if you see, one of the mechanism is apoptosis of the cell death is there. So in the humans, there is a probable link of esophageal cancer. We have seen IARC carcinogenic to be, it has been classified. Occurrence of this fumonisin B1 is correlated with occurrence of higher incidence of esophageal cancer in the regions. So particularly in South Africa, in China, as well as in the Italy. Means, you see, it is covering completely entire geographical population. If you see African country, China, that is Middle Asia, as well as Italy, that is European countries also, you can find this particular type of fungal poisoning is there. Okay. So in addition, what is there in the textbook only? Clayson, we are reading Clayson. It's a very good quick revision, this particular slide. A possible case of acute exposure to fumonisin B1 involved 27 villages in India. So there was an outbreak, epidemiological outbreak was there because of this contaminated corn. So those 27 villages in India, which took place in early 19, I'm not remembering, early 19th century only. So you go back and you can go back historically and find this incident. where consumption of this unleavened bread made from moldy sorghum or corn caused transient abdominal pain. And another symptom that you can see is borbogymus. What is that means? It is nothing but you'll have some this gurgle in the case of stomach rumbling will be there. And another thing is diarrhea. Okay, whenever you contaminated food poisoning, diarrhea is one symptom you will find it. So all those people, there was no mortality. They were recovered this particular population, but there was outbreak was seen in this particular 27 villages in India. Next, if fuminogens can cause neural tube defects. So till now we were discussing about all other type of this, but now we are discussing about CNS, neural tube defects in experimental animals may also have role in the case of human cases. IARC, I told you, International Agency for Research on Cancer, evaluated the cancer risk of these fuminosins in humans and classified them as group 2B. Okay, so pause here, read about this IARC classification. I have already given here also. Some people might have been already aware. So there are some examples are also listed here under this particular diagram. So pause the video and common examples you can see which will fall under group 2A, group 1, group 2B as well as group 3 when it is animal carcinogen and when it is human carcinogen, both animal and human carcinogen, the classification is given here. Okay, there is a need. What do you mean by group 2B? For example, we'll read here. There is an inadequate evidence of carcinogenicity in humans. So there are not enough epidemiological or population studies are not there where it can be proven that this particular thing, fuminosin toxins, there is a correlation with the cancer. Okay, but there is a sufficient evidence of carcinogenicity in the animals. So if you look into a typical 2-year rat and mice study, probably in that they have found that there are tumors are there. But if you look into population studies, epidemiological studies, or cross-sectional cohort studies will be there. No, in that probably they could not have established a relationship, a strong cause and effect relationship between the cancer as well as the fumarocins. Okay, fumarocins target different organs in various species, but underlying mechanism is disruption of this lipid metabolism. This is very very important that you have to remember as a mechanistic toxicology. Inhibition of ceramide synthase, an enzyme integral to the formation of complex lipids for use of this membrane. So whenever you have phospholipid bimembrane, every cell, no, not every cell, prokaryote, eukaryotes, the cell is different. But if you look into our human cells only, phospholipid bilayer will be there. So for the formation of this lipid, you need a lipid metabolism cycle will be there, lipid formation, fatty, this thing. So in that, this integration of this complex lipids into the membrane, this particular step, inhibition of the ceramide synthase, this particular fuminosin is taking, is doing that mechanism is there. Okay, so pause here, read about all this IARC classification which is given by this particular taken is from World Health Organization. Okay, so in addition, we will also be reading in the class and ninth edition only, page number 1348, one table is there, table 2711. So this 2711, it contains selected mycotoxins produced by various molds and some of the effects and comorbidity potentially contaminated. So we will see what are those one. First one, let's stick to the question. The question is with respect to fumonosins. So in that, as I told you, different fumonosins are there. B1, B2, B3, B4, A1 and A2. Most of the literature is on B1. So whatever options we have seen that fumonisin B1 is the potent one. So in that also see you can verticillioides, but formerly it was known as moniliforme. Like that you have to remember, and it is causing kidney and liver cancer. Contaminated corn. Okay, commodity is contaminated, corn is contaminated. Okay, let's see other options. Let's not just stick to this particular question. With respect to table 2711, also read other things, other food contaminants and with fungal toxins, which were previously asked in the DABT questions or might in future also ask. So first one is aflatoxin. Second one we'll read about aflatoxins. Aflatoxins also very very important. So whenever the food like corn, peanuts and milk, if they are contaminated with this particular mycotoxins, so the toxin which is released, mucotoxin which is released is aflatoxins. In that also you have B1, B2, G1, G2 and M1. Okay, so what are they causing? They're causing cancer. Aflatoxins are causing cancer and the source is aspergillus flavus and their metabolites are also there. So pause the video and read or go back to textbook and read. And next one we are reading about whenever the cereal grains or corns are contaminated with one and trichothecenes are released. So trichothecenes, that is T2. Some questions were asked on the previously in DABT, they have asked on the trichothecenes. So we have deoxy 11. This is also very important. If we look into the mycotoxins, no, in the European countries also, they have listed in the food products, no, these are the commonly found toxicants, fungal toxins. So they are they these particular type of toxins, they need to be regulated with certain limits. Okay, so deoxylavinol is also there in that particular list. Okay, you can go back and check what are the regulatory standards in the case of European region. Okay, the source is fusarium and mycothecium. Okay, and it is going to cause blood toxicity, that is hepatotoxicity, meningeal hemorrhage in the case of brain. Okay, they are going to interrupt your meninges layers, nervous disorders, necrosis of the skin, hemorrhage in the mucosal epithelia of the stomach, intestine, emesis, feed refusal, and immune suppression. So these are the things which trichothecenes. So previously T2 or trichothecenes you will get in your option in the exam, or you will also come across deoxynivalenol. So this also we will come across in your exam. I'll come across where I have some questions from other years, because till now we are discussing year 2000. That some other year question papers might be there, in that they might have come across this one. Next one is zearalenone. Zearalenone, whenever the corn or grain is contaminated, again it is coming from fusarium only. It is going to affect your estrogenic effects. So zearalenone also they will ask, one of the options you will be having in the exam. Then we are ochratoxin. Ochratoxin is also important. Grain, peanut, grapes, and green coffee, whenever they are contaminated with fungal toxins. And the source is Aspergillus ocellaris, that's why the name ochratoxin. And other Penicillium vericranium is also given here. So it is causing endemic nephropathy, right? And it is causing carcinogenesis. Okay, then next we have ergot alkalides, grains, coming from Claviceps purpurea, and main thing, it is causing ergotism. One more questions you will come across whenever you're reading about ergotism: Saint Anthony's fire. So there is one classic example historically in the European countries it has taken place. So you read about that also. And what else are there? Potolin, which is coming from apple. Whenever the apple or apple products are contaminated, the source is again Penicillium putillium. Probably that is the reason the name is Potolin. So in the case of DABT, there used to be a Twitter group used to be earlier. So in that I have around 50 to 60 questions from that from the seniors. So one question was about with respect to apple toxin, toluene. Whether it came in DABT, I am not, but in the study group this question has come. So it is there, the major adverse effects it is causing cancer and liver damage. Okay, so pause here, try to read other examples. Okay, very good table, because on the in the single table you have got all the fungal toxins, mycotoxins. So thank you so much. Be curious. For any questions, you can always write a comment to arya.priyanka18@gmail.com. All the best for your exam.